Module 2, Enzyme Complexes, Assays, Purification, Reconstitution, and Regulation. Approximate 3000 plus words. Introduction. Welcome to Module 2 of the course. In Module 1, we covered the fundamentals of enzyme kinetics, catalytic strategies, and the principles of allosteric regulation. In this module, we will shift our focus to the structural and experimental aspects of enzymes, including multi-enzyme complexes, enzyme assays, enzyme purification, reconstitution, and the regulation of enzyme activity and synthesis. These areas form the cornerstone of applied enzymology and molecular biology, particularly in biotechnology, medicine, and pharmaceutical development. Section 1, Multi-enzyme complexes. Multi-enzyme complexes are associations of multiple enzymes that catalyze successive steps in a metabolic pathway. These structures are often physically connected, allowing substrates to be passed directly from one active site to another. 1.1 Definition and characteristics. A multi-enzyme complex consists of several enzymes bound together, either tightly or loosely, often within a single protein framework or cellular organelle. These complexes are usually involved in enhancing reaction efficiency, reducing diffusion time between intermediates, coordinating regulation of multiple steps in a pathway. The close proximity of active sites facilitates substrate channeling, whereby intermediates are directly transferred between enzymes, minimizing loss and side reactions. 1.2 Examples of multi-enzyme complexes. Some classic examples include pyruvate dehydrogenase complex, catalyzes the conversion of pyruvate to acetyl-CoA. It consists of multiple enzyme subunits working in tandem. Fatty acid synthase complex, a large multifunctional enzyme that synthesizes long-chain fatty acids through repeated cycles of condensation and reduction. Ribosome, though not an enzyme complex in the traditional sense, it acts as a molecular machine facilitating protein synthesis with high efficiency. Multi-enzyme complexes are essential for increasing the fidelity and speed of biochemical pathways. 1.3 Functional advantages. The benefits of multi-enzyme complexes include minimization of side reactions, enhanced control over pathway flux, simultaneous regulation of multiple enzymatic steps, structural stability and protection from degradation. Biotechnologists are increasingly designing synthetic multi-enzyme assemblies for applications in industrial catalysis and synthetic biology. Section 2, Enzyme assays. An enzyme assay is a laboratory procedure used to measure the catalytic activity of an enzyme. It is one of the most essential tools in enzyme research, allowing scientists to characterize, compare, and quantify enzymatic reactions. 2.1 Types of enzyme assays. Enzyme assays are generally classified into two broad categories. Continuous assays, these monitor the enzymatic reaction in real time. The change in absorbance, fluorescence, or conductivity is measured continuously. Discontinuous assays, in these, the reaction is stopped at specific time intervals, and the concentration of the product or substrate is determined after stopping the reaction. Common formats include spectrophotometric assays, based on absorbance changes due to substrate or product. Fluorometric assays, use changes in fluorescence to track reactions. Radiometric assays, track the incorporation or release of radioactive atoms. Chromatographic assays, separate and quantify reaction products using chromatography. 2.2 Considerations in designing an enzyme assay. To obtain accurate and meaningful data, several factors must be considered. Choice of substrate should be specific to the enzyme. Buffer system maintains optimal pH and ionic strength. Temperature control, as temperature significantly affects enzyme activity. Time points, reaction time should be sufficient to measure initial velocity but short enough to avoid substrate depletion. Additionally, enzyme concentration must be low enough that the reaction is linear with time. 2.3 Applications of enzyme assays. Enzyme assays are used for characterizing new enzymes, determining kinetic parameters, Screening for enzyme inhibitors. Quality control in industrial enzyme production. Diagnosing metabolic diseases. Assays are also critical in drug discovery, where enzymes are tested against thousands of compounds to identify potential drugs. Section 3. Criteria for determining purity of enzymes. Purity is a crucial parameter when studying enzyme properties. Impurities can interfere with activity measurements, structural studies, and downstream applications. 3.1 Common purity metrics. Several metrics are used to evaluate enzyme purity. Specific activity, defined as enzyme activity per unit of protein. As purification proceeds, specific activity typically increases. SDS page, electrophoretic separation of proteins by size. A pure enzyme will show a single bond. Isoelectric focusing separates proteins based on their isoelectric points, useful for detecting isoforms or charge-based impurities. Chromatographic profiles, a sharp and symmetrical peak in HPLC or gel filtration indicates high purity. 3.2 Enzyme homogeneity. Homogeneity goes a step beyond purity. It means all enzyme molecules are structurally and functionally identical. Techniques like mass spectrometry, sequencing, and structural analysis are used to confirm homogeneity. 3.3 Importance of purity. High purity enzymes are essential for crystallography and structural determination. Industrial use, where impurities can affect product quality. Clinical diagnostics, to prevent false results. Research reproducibility and data integrity. Section 4. Enzyme reconstitution. Enzyme reconstitution refers to the process of restoring enzymatic activity after the enzyme has been denatured, isolated, or modified. This process is critical in studying enzyme mechanisms, refolding proteins, or restoring multimeric enzymes. 4.1 Reconstitution of apoenzymes. Some enzymes require cofactors, such as metal ions or coenzymes, to become active. An apoenzyme is the inactive form of an enzyme that lacks its cofactor. Reconstitution involves adding the missing cofactor to the apoenzyme, thereby restoring activity. 4.2 Refolding denatured enzymes. Recombinant enzymes produced in bacteria often form inclusion bodies, which are aggregates of denatured proteins. Reconstitution involves solubilizing these aggregates and refolding them into their active conformations. This process may require dilution in buffer, use of reducing agents, addition of chaperone proteins or additives. Success is typically confirmed through activity assays and structural studies. 4.3 Multimeric enzyme reconstitution. Some enzymes are composed of multiple subunits. Reconstituting such enzymes involves assembling the subunits correctly and ensuring proper orientation and interaction. Reconstitution studies help identify the role of each subunit, map active sites, and investigate intersubunit communication. Section 5. Regulation of enzyme activity and synthesis. Enzymes are tightly regulated to ensure metabolic efficiency and cellular homeostasis. Regulation can occur at the level of enzymatic activity or at the level of enzyme production. 5.1 Regulation of enzyme activity. Activity regulation ensures that enzymes function only when needed. Major mechanisms include allosteric modulation. Activators or inhibitors bind to allosteric sites, changing enzyme conformation. Covalent modification, addition or removal of chemical groups, e.g., phosphorylation, alters activity. Proteolytic activation, some enzymes are synthesized as inactive precursors and activated by cleavage, e.g., digestive enzymes. Enzyme inhibitors can be reversible or irreversible. Reversible inhibitors bind non-covalently and can be removed, while irreversible inhibitors permanently inactivate the enzyme. 5.2 Regulation of enzyme synthesis. Long-term regulation involves controlling the expression of enzyme encoding genes. Mechanisms include induction, increasing transcription in response to a substrate or signal. Repression, inhibiting transcription when an end product is abundant. Post-transcriptional regulation includes mRNA stability, translation efficiency, and protein degradation. Such regulation is crucial in adapting to environmental changes, developmental cues, and stress responses. 5.3 Examples of enzyme regulation. In the lac operon of bacteria, the presence of lactose induces the synthesis of beta-galactosidase. In cholesterol biosynthesis, the enzyme HMG-CoA reductase is regulated by feedback inhibition and controlled degradation. Hormonal regulation in mammals adjusts enzyme levels through signal transduction pathways. These examples illustrate the diversity and complexity of regulatory networks in cells. Conclusion of Module 2. In this final module, we examine the structural and functional complexity of enzymes beyond their catalytic action. We explored multi-enzyme complexes, which streamline metabolic processes, and enzyme assays, which are critical for characterizing enzymatic function. We also discussed the criteria for purity, reconstitution techniques, and regulatory mechanisms that govern enzyme activity and synthesis. Together with Module 1, you now have a comprehensive understanding of enzyme structure, function, regulation, and analysis, essential knowledge for careers in biochemistry, biotechnology, pharmacology, and molecular biology.